Welcome, everybody. Let's get the ball rolling. Welcome to Brain Imaging. I'm really looking forward to these next couple of months. I'll be trying to teach you all I know, or at least as much as we can cram into these 2 months about brain imaging. What we'll do today is run through a brief intro. What is the structure of the course? And then I'll just try to start giving you an idea of what it is that we're having to deal with if we want to sort of distill mental processes from someone's brain as they're inside an MRI scanner. There's a lot of ins and outs there, so let's get going. Yeah. Immediately this thing stops working, but so who am I? I'm Thomas. If you have questions, you can email me. That's my email. And you can also always DM me on Discord if you have a pressing matter. I have it on my phone, so I can just reply to you as soon as possible. It's my experience that works a lot more fluently than Canvas messaging. Yeah, let's use that as a discussion board during the course. There are specific channels there also. This is me also, this is my brain. This is also me. That's my visual maps in the back of my head that we've mapped using functional MRI. So one of the things that you might learn in one of the optional advanced notebooks is to create maps such as these from a data set. So that we give you a data set and you'll learn to construct these types of maps. So how do you go from putting people in the scanner to a result like this is what this course intends to teach you. Yeah. We do a bunch of work where our research is really focused on using the MRI scanner to understand how the human brain processes information, and we try to use it as a tool to understand how our minds work. We have two TAs that I don't think are here right now because miraculously both had dentist appointments at just this moment, but you will see them during the practicals, for example, and they will be present at other lectures. Ron and Shifan are PhD students in the lab. They're both conducting their own experiments and analyses on this type of thing that you see at the bottom of the slide. And specifically, we're usually interested in analyzing the visual system, and that's why you're also seeing the back of the brain there. We do a lot of different things, so we collaborate. This is just to introduce the type of research that we do so that you have an idea of where we are coming from, right? We do a lot of fMRI like development research at the Spinoza Center that's in the city here, where we have our scanners. We collaborate there. We are also part of the cognitive psychology group here and of artificial intelligence, and we have a bunch of US collaborations that might not be interesting for all of you, but if you want to do, for example, an international internship, I just want to highlight that if that's your interest. We have a broad international network where, you know, you can do an internship somewhere else if that's what you want at some point. So that's just about me. What about the course, right? I'm sure you're all anxious to learn exactly what I expect from you in terms of what you're supposed to do during the course. Now, I'm sick and tired of doing things over Zoom. I think the world is much better in 3D. That's why I'm forcing you all to be here. I'm hoping that we can have these lectures in a much more interactive way than you can do over Zoom. Really, to me, increases the sort of intensity of the teaching experience, and I've noticed that it also helps people just in terms of participation. Oh, that's Shifan, by the way. He's one of the TAs. So lectures face to face. Now, of course, I do want to be forthcoming in terms of if you have a dentist appointment, say, right? If you're able to not come for a given lecture, just let me know over Discord. I can send you a Zoom link. I am sharing the screen over Zoom. This is just a broadcast for those people that cannot attend any given lecture. It's not being recorded, right? So really, I'm trying to get you all to participate in the lectures in this way. You do have the PDF, so you do have all of the static content available to you for study later on, but I think it helps really if we all are here. Now, on the other side, what we ended up doing when Corona hit was we migrated all of our practicals to Google Colab, because then we could run all our practicals online and everybody could participate. And that I did really like, because it gives you as students the freedom to just, take up a practical and work on it if you are a night person in the middle of the night, if you like. And so we're hosting these practicals still on Google Colab. They will become available throughout the course. There are 3 practicals. The first one's online already. It's just a very simple introductory notebook so that you can get a handle on how to work with these notebooks and how to work with the basics of fMRI data. So consider that mostly a Python refresher that's online now. So that's going to be easy for you. Most of you have seen that one of the prerequisites of the course is Python, well, not fluency perhaps, but experience definitely. Now we do have 3 practical instances. They're going to be on Wednesdays, the 2nd, 4th, and 6th week of the course. They're going to be right after this lecture on the Wednesday, and that is when Ron Schufan and I will be here to help you with any questions that you have regarding the practicals. So we will be able to help you face-to-face with any questions. Any other questions that pop up, for example, when you're burning the night oil, working on these at 4 a.m., you can send me messages over Discord. I will be asleep, but hopefully at least. So that's how we try to give you the freedom to work on these practicals whenever you like, and we are able to help you whenever we can. So what will you learn during the course? What I'm going to try to show you today already is a bit of a feel for what fMRI data are composed of. Right? You put something in the scanner, you get out data. What does that feel like? And so I'm going to teach you a bunch of theory about what you can and cannot learn, for example, from an fMRI experiment. Things like how do you design an experiment are really important in that context. And when you do an analysis, of course, you get results and then you have to interpret them. So how do you do that? Right, so that's really the sort of knowledge part of the course. But in my experience, fMRI, there's a lot of practice in it too. You end up spending a lot of time designing an experiment, conducting an experiment, and then analyzing the experiment. So it's not just knowledge that you need to learn during a course like this. A very important component is also this know-how, right? And for that, we're using these practicals, right? So in the practicals, we'll really allow you to work with MRI data that we've gathered ourselves or that we have downloaded from the internet, which isn't even illegal. And then we'll analyze fMRI data in like the most standard way to give you a basic understanding of how, when you read a paper, for example, what are the data like? So we'll do that during the practicals. And we have some optional notebooks also that give you an insight into more advanced analyses that you can try out for yourself. Yeah. So this, 2 lectures a week. It's not going to be enough. I have way too much to tell you, but we'll try to compress everything. And I hope that we can have these discussions face-to-face about any topic that you find interesting, right? So if you have a specific thing that you want to talk about and you want to see the brain results for example, then just let me know and we'll try to see if we can fold it into the course. So we have these 3 practicals. Oh, sorry, yeah, I'm skipping something. So we have a bunch of guest lectures, for example, on the MR physics side of things, right? Well, how does a scanner work, that sort of thing. We have a bunch of guest lectures on that sort of thing. And because I want to sort of show you very novel stuff, I also have segments of a YouTube playlist by a bunch of other researchers from the US where they explain the basics, right? So you can always go back to these 10-minute, 15-minute knowledge clips made by others that give you like a different view on the same course material. But it's my impression that that really helps different students understand different perspectives on the topics. Yeah, so the practicals are 3 of them. When we meet, there are Perusall reading assignments. They'll be you'll be notified as assignments on Canvas, and you'll see them in Perusall. And we have the Discord community thing server running. So everybody, you can just click on the link, I think, in the syllabus in Canvas, and that should bring you to Discord. So please make sure that you're on there too, so we can start discussing things there. Yeah. Cool. Now, I'm not very grade oriented, but I know that some of you are. So how do you build up a grade in this course? We have an exam at the end of the course. That's the majority of your grade. That's the major proportion of your grade is determined by the exam. And it will be mostly multiple choice, but there are going to be some essay questions too. They're mostly bonus questions, right? Where you can decide to either shine or not in these essay questions. You'll see multiple choice questions throughout the course. Every module that we have on Canvas has a quiz attached to it. And so you'll get a feel for the types of questions that we're also going to ask during the exam. So you'll be able to practice those questions also during those quizzes. So I'm already giving you that. Exam materials, the lectures that we'll be doing here, all of that content, the PDFs, for example. The articles that we discuss and read on Perusall are also course material that you're required to know for the exam. And selected YouTube modules, like the YouTube modules I just talked about, the ones that you can peruse at your own leisure. The concepts that overlap with my lectures also are part of the materials. And then finally, so we have some quizzes at every end of the module just to give you an impression of what you're supposed to know. And then we have reading participation that's graded by Perusall, which is also a small portion of your grade. Any questions? Because I'm going to start talking about fMRI. Yeah, so any quick questions about what I expect from you? If you want to know what you can expect from me, we can go into that too. No questions? Excellent. Everything is clear. Or it's just too early to ask questions. Okay, so what I want to do in the remainder of these 1.5 hours that we still have is to show you like a broad overview of what our course is going to give you. There's the first PDF that you're supposed to read on Perusall. You're going to be able to see it, I think, today already. You can just link to it. It's also very much an overview article. Like, what can you do with fMRI? So in this first lecture, in this first reading material, this first week, we're just going to go into these basics, like very broadly, what can we do with fMRI? What can we not do with fMRI? And so today I'm going to go through a bunch of these topics really quickly, and most of these we'll dive into in more detail in later lectures. Yeah. Who of you have ever been in an MRI scanner as a subject or patient? That's about 10%, I think. Yeah. Excellent. What was it like?
Scary.
Scary. How so?
I just didn't like not being able to get out of it.
Ah, yeah. Yeah. Yeah, there's a bit of pressure to actually press the button, right? Yeah. Anyone else want to share an experience that they...
Too loud.
Too loud? Yeah, yeah. So this is what we do. We put people in a tube, in a large tube, right? They're in there, only their feet are sticking out, so you can sort of see their toes wiggling, but they're inside a big sort of container, and it can be really claustrophobic. You are not supposed to move. So you have this padding on your head. You're not supposed to move. If you do, then I'll show you what happens in terms of the results, and it's not good. And so you can get really claustrophobic because, you know, you're stuck there, right? At the same time, whenever the scanner is acquiring data, it is making a shit ton of noise. And in the scanner that we use most, this can go up to like 110 decibels, which is, you know, worse than your favorite dance floor. And so you definitely need a lot of ear protection to even make it bearable. Yeah. And so we need to, we're really asking something of people when they do this. And as a researcher, we also have the sort of obligation to make sure that people are as comfortable as we can make them in the scanner. If you want to be a subject after hearing all of this terrible news, there is a sign up at the Spinoza Center website. I'll put it on Canvas and Discord. It's a nice, interesting experience. So if you want to be a subject, you can sign up for specific experiments on Sonar. There are a few researchers also part of this course. Rebecca, you're still scanning, right? Some of these.
Yeah, but I'm looking for boys of 16 and 17.
Any boys? No. Nah. Yeah, so specific research sometimes requires specific populations of subjects. There are other studies you can participate in and help us and the world, right? Science, yay. So what is this fMRI, right? That we ask people to perform this horrible thing with all this noise. Well, it's a really, really big research field nowadays after it was invented. It's really, really exploded in terms of how many researchers conduct this type of research. Also, how many people have to endure this sort of torture. What I'm showing you here is a graph of how many publications were published every, I think, month or so over time. And you see that fMRI was sort of invented around the 1990s. We'll go into this history of how fMRI was invented. And you see that it's really increased tremendously over the last years. And it's starting to top out a bit because you can only do so much science. I think it is. There might be other reasons why it's leveling off, and we can talk about that later on in the course. What I think is really cool, and what's really driven me to start doing fMRI, and what I think some of you might have chosen this this course for, is really that when you read about what fMRI is, it's how we place mental processes inside the head, right? So we know a love area in the brain. So how do we get that, right? So this is really sort of the magic of fMRI, that we can start to place the mind inside the head. And really in popular science, that's really what's happened also as a result of this boom in research. So our goal here is to show you how you do this type of research, right? And sort of also lift the veil a bit and make this a bit less magical, because it's known that when you just show people a blob in a brain, they're more likely to be convinced of any text that's right next to that image of the blob in the brain, right? So it can serve as a sort of cheat also. And what I think you can definitely get also from this course is that that we lift that veil, that it becomes less of a sort of magical thing when you see a blob in a brain. You know exactly how to create these blobs. And so you can be much more critical of whenever you see something in popular science literature, for example. You won't be able to be fooled as easily, I hope. Maybe by me. Yeah, so why this boom, right? There's a big driving force behind this huge boom in papers, right? In the research that people have been able to do. And that's mostly because people have these scanners anyway. They are in hospitals because they are also clinical machines. So most of the MRI scans that are done in the world are made of knees, of livers, of that sort of thing. And it's just a coincidence that we can also do fMRI and actually do cognition brain research using the same machines, right? So it sort of comes for free, and it means that any city that has a hospital, you could in principle also conduct this type of research. And that's a really liberating thing for scientists, of course. Now, where is this field going? What are the new developments? This first paper that you'll be reading on Peruso really goes into what the recent developments are. How are we using the MRI scanner for measuring the brain? What types of possibilities do we have? And what is the future for this field? So it'll be a bit of a hopscotch through the different possibilities reading this paper. So perhaps you feel like, I'm just scratching the surface here. Many of these topics we will go in depth more when you read this. And again, if you have any questions when reading this, just consult me over Discord, for example. So why fMRI? Right, so we could also be talking about EEG. There's actually a different course in the Research Masters for Psychology on EEG. You could also equally do like animal neurophysiology. So why really do we want to do fMRI research? And that has to do with where fMRI is in this graph. So what is this graph? Well, it's listing different measurement techniques that we have, different ways of peering into brain activations, understanding brain activations. And it scales, there's two different scales. One is temporal resolution, so that is how fast can you measure and how fast do the processes that you're interested in transpire, right? So on the left, you see very, very fast processes, and on the right, you see very, very slow processes. From milliseconds here to months. And the other dimension here is spatial resolution. So that's the level of spatial detail that we're interested in. And so we can go all the way to the synapse level, for example, to the molecular level here at the bottom. But we can also look at like what happens throughout the brain. So taking a whole brain, for example, as a measurement, right? So we can choose if we have a specific type of process that we're interested in. This sort of diagram gives us a way of deciding what techniques should we use. If we're interested in synapses, maybe not use fMRI. So where is fMRI here? You see that is this very, very large blob in that region of this space. You can't do single cell electrophysiology with an fMRI machine. That's clear for other reasons also that I'll get into in a bit. But you see that it is very large, right? It does span a bunch of these ranges. You can go to different levels of detail with the fMRI, right? Another reason that it's so popular is that it's non-invasive. For many other techniques that you can do in humans, such as, for example, PET imaging, you have to inject people with a contrast agent. And in the case of PET, it's not even invasive in terms of that you have to put something in their bloodstream, but it's even a radioactive substance that you have to put into their bloodstream. You can imagine that you can't do that over and over and over. I've been in an MRI machine for over 200 times. That would not be possible for PET imaging, for example, just because of the radioactivity load. Yeah. It's not just a very big part of this space already. It also it's in a good location here. It isn't right there, it's right there. What that means is that we can look at things that happen on the second scale. And if you sort of think, what's an interesting scale to look at cognition? It's around the second scale, or maybe things that are longer, right? So if you're paying attention to what I'm saying, the meaning of the words that's unfolding, or that I'm speaking as it unfolds, is something that you're tracking over seconds, right? So it's an interesting time scale even to be looking at. At the same time, we can measure the whole brain at a level of detail that allows us to really pinpoint specific locations, right? So this combination of time and space that fMRI provides is a really, really cool sort of focal point to have if you're interested in human cognitive processes. We can look at entire lobes, we can look very, very specifically. And as the technique is evolving, it's actually becoming faster, so we can measure faster processes with the same scanner, because a lot of developments in terms of AI are helping us scan faster, for example. And similarly, these same developments and different developments are allowing us to scan more precisely. And I'll get into that too. Like, how are we starting to use the scanner to be able to look at, for example, layers of the brain instead of just whole regions of the brain, or look at like sub-second responses in the brain. So the evolution of this technique is also helping us become more powerful scientists. Yeah. There's also a limitation, of course. Anyone think of a limitation, that the sort of experiment that you just shouldn't do if you're interested in? So fMRI would be really bad if you're interested in fill in the blank. Movement. That's an excellent. It's a very, very good point. Yeah, so you have to be still, right? So I don't know who said it. You would. Oh, there you are. Yeah. Indeed, keeping people still is a major downside of fMRI. And it really constrains the type of experiment that you can do. Yeah. I'll show you later on how horrible movement is for MRI. It's the padding, for example, and the instruction to not move. They're not there for nothing. They're really, really prominent feature of what you cannot do with fMRI. Exactly. Anyone else? Another thing that you cannot do with fMRI. If you look at this square.
Study the encoding of a specific neuron.
Yeah, exactly. So you cannot go to that level of detail. You can go to a level of detail with non-functional MR imaging that's, you can sort of resolve like clusters of neurons, but you need a dead person's brain to do it, because you need to scan for like 24 hours or so. Yeah, so that's not really where we want to be. Another thing that you might not be wanting to use fMRI for is if you're interested in, for example, very fast working memory, like or what happens around the time of an eye movement, right? That happens in 300 milliseconds. And you see that 300 millisecond range would be like right at the boundary there. Yeah. In that case, if you're interested in that sort of process, you move to a different modality, right? We have to be flexible as researchers if we have a specific question. For example, this fast memory type thing, or eye movements, you go to EEG and MEG. And there are beautiful developments in those fields too that I don't care about because I'm an MRI person. So we can talk about that outside of this course. This course is really focused on fMRI, and it's plenty of topics to, yeah.
What's the difference between like fMRI and just MRI?
Ah, yeah, that's a good question. They're in principle the same thing. It's just that in fMRI, you don't just take an image of the brain once, where you get a picture of the anatomy, but you do it multiple times in a row, where you can start to look at changes over time, and that makes it functional. Right? So then you can, for example, start doing an experiment where people are shown specific types of images at a specific moment in time, and you can look at how the fluctuations of these images that you're taking depend on what you're presenting, and that makes it functional. So that this allows us to really link what we think the brain is doing to these sequences of images as they unfold over time. Yeah, I'll go into this difference in more detail later also. Yeah, but you can, yeah, go ahead.
Yeah, I was thinking, so is fMRI then multiple MRI scans?
Exactly. Yeah, yeah. I'll try to find some sound clips. I'll try to record them this week, where I also sort of let you listen to the difference that you have between anatomical sequences and functional sequences. There are very distinct differences that those of you that have been a subject, for example, in an fMRI experiment will be familiar with. Okay, now I don't know what happened with that. There it is. Yes. So this functional MRI. I'll get into the functional versus anatomical thing in a bit. But one thing to be very aware of, and this is something that let's take a step back. This one thing that I'm gonna be teaching you is fMRI is mostly nonsensical. That might sound weird from the teacher right now at this point. But what you have is a measurement technique that is very noisy. It's not measuring exactly what you want it to measure. So what we would want, of course, is for our brain activations as we get them from our analysis, we would want them to show neural activations, really the neurons firing, right? That would be awesome. But we don't have that. In essence, what we're measuring with the type of fMRI that we're doing is blood changes, stuff that happens in the blood. Now, it makes sense that if a bunch of neurons are active, then the blood will follow, right? So there is a link between the blood flow and the neurons firing, the thing that we're actually interested in, but this link is indirect. Right? We are not performing a direct neural measurement. And this is just one of the things where it's important to realize that you may think that you're looking at something very specific, but you're seeing it through different lenses. So in this case, what you're always looking at in terms of fMRI results is this blood-soaked lens. Everything that you're seeing is through a blood response. This is also what limits our temporal precision, for example. Yeah.
Is there any way to tell whether the blood is going to like inhibition or excitation?
Yeah, that's a very, very, very good question. And the jury is still out on that. So there's a lot of literature, also in animal studies, that are looking at whether it matters for the bold response, whether the cells that are firing in a specific location, whether they're excitatory or inhibitory. And in principle, you have to maintain the rule of thumb. We don't know, right? So it could be 100% of the inhibitory neurons firing or 100% of the excitatory neurons or 50-50. They could give you the same bold response. And there might be, it might be stronger for excitatory or inhibitory, but we don't know the ratio, right? So people are trying to find this sort of ratio and be really specific about it. But all we have is a number at a specific location in the brain. We know that it depends on the blood. Right? So caveat number one, fMRI may produce these beautiful images of a living human brain. And it's good because living brains, they have blood flow and all of that. But you are looking at blood, right? It's an indirect measurement of the thing that you're interested in. And you always have to keep it in mind, right? Because it could be something completely different than what you think it is because of this. Yeah.
So how do you do MRI on that?
Yeah, so you cannot do fMRI.
Oh.
Yeah, so what you can do is you can do very detailed anatomical MRI. And there you create one three-dimensional image, but you just pool a lot of data. So you get beautifully precise measurements, beautiful images you can get from postmortem brains. But of course, yeah, they're dead. So you can show them an image, but they won't respond. And usually what you also do is in a postmortem brain, it's just a part of the brain that you're scanning. So you can get really, really precise. Yeah. Yeah, that would still, and that's also big benefit is that people don't move, right? So, yeah. You had a question?
So what is MRI measuring? If not blood? You take it of a knee, for example.
Yeah.
What are you actually measuring?
Yeah, so we'll, on the 22nd of this month, Nikos Priovoulos, who is a colleague of mine at the Spinoza Center, he's an MR physicist, and he will tell you all about the different mechanisms that really have to do with like the nucleus of the atoms and how we're sort of manipulating the nuclei of atoms in order to produce these signals and images. So he'll get really into the nitty-gritty, but the major idea is that our brains are composed of a lot of water, and most of the imaging that we do has to do with this water. What we do is we are manipulating people with this huge magnet, which is a scanner. And what we're manipulating is not their sort of neurons or whatever. It's really the magnetic alignment of the protons in the water in their brains. And so, yeah, it's all very quantum mechanics in the background. But you can think of these protons as sort of spinning tops, right? So And normally they spin in all sorts of directions, but if you apply a main magnetic field, a very strong magnetic field, they all sort of start pointing in the same direction. And then the idea is that this sort of magnetizes the tissue. You don't notice anything because it's happening at an subatomic level. But in total, this means that there's a sort of tension in the tissue, a magnetic tension that you can then start playing with. And so what the scanner does is it plays what we call sequences and thereby manipulates the magnetism locally. And by interacting with radio waves, you sort of get the images. That's all I can say right now, but it will become much clearer, and I'll go into it in a bit later. Okay, so caveat number 1, right? Blood, not neurons. Very important. Now, it gets worse. This is not just, blood, right? So whenever a neuron fires, I get X blood, we're good, right? No. There's an entire sort of metabolic sort of cascade of events that happens after neurons fire. So neuron fires, and the neuron, of course, is embedded between all sorts of other cells and blood vessels and what have you. And so this whole physiology around that neural activity then responds. There's a whole cascade of different events. The tissue itself moves, the blood comes in and flows out. An incredibly complex machinery that just provides glucose and provides oxygen to the tissue so that the neurons can fire. And it's through this lens, I'm not going to go into the specifics of these elements right now, that's for later, but through this, that's the response that we're measuring, right? So we might be interested in this neural response that we try to manipulate in our experiment. What we end up measuring is a Bode response. And for example, if people are holding their breath, right, this has all sorts of effects on the CO2 pressure in the blood and the oxygen level in the blood that will give you a completely different response than if people are just breathing normally. Now, what we do in a standard way, and this is caveat number 2, we just assume that the relation between this neural response and the response that we measure, that it abides by a standard shape. We call this a standard hemodynamic response function. We'll get into all of that later too. And rest assured, this is a caveat number 2 because this stereotypical shape that we assume holds across subjects, across brain regions, it makes no sense whatsoever. The shape of this response varies across individuals, across age, across brain regions, across whatever. And so one sloppy thing that we generally do in fMRI is we assume that it's all the same, and then we see how far we get. Okay. Like we do knowing that we're not measuring neurons. We just say, we're measuring blood. Okay, so let's see what happens, right? And that's the eternal state of the field. Maybe it's a science in general sort of thing. You're just taking this and seeing how far you can run with it. Okay? But you have to be aware that that's what you're doing. Yeah. So caveat number 2. We really don't know what we're doing. That's caveat 1 and 2 together. There are more caveats. Right. So just this slide, and then we'll go to break. Like I said, all of these hospitals, they already have a clinical MRI machine. And over the second half of the 20th century, this device was developed to produce more and more different types of images, and improvements in things like electrical engineering and computer science have really made this better and better. It's really also like the forefront of science. There are multiple Nobel Prizes associated with MR imaging. Just the idea of being able to produce images with a big magnet. We'll wrap your heads around it during the course, but it's pretty insane that you're able to do that as a monkey species that we are, right? In any case, this machine, it can make nice images of knees, for example. You can also look at brain activations. You can also look at fiber bundles running through the brain. So this machine is very, very versatile. It's all about how you use the magnets and the radio transmitters and the radio receivers to generate images. You can also look at the chemical content of the brain, for example, as well as spectroscopy. Just another way of using the scanner. So it's a very, very versatile machine. And what's the difference between all these different types of measurements? It's how we use the scanner. The word that we use for that is sequences. So a sequence is just a program that you run on the scanner that allows it to produce a certain kind of image. Yeah? And similarly, you can think of this as like it's hooked up to a computer, right? If you have, I don't know, a scanner attached to your computer, right? You document scanner, you can just set it in a program. You can set what's the resolution that I want to scan this at, right? Is it like a photograph or is it like a black and white document? You just run a program and it scans differently. Similarly, for an fMRI scanner, it's just like orders of magnitude more complicated than a document scan. Now, one major thing that, like the major element of the MR scanner is the huge magnet that's at its core. That's the thing that you're lying inside of, the thing that's causing this incredible amount of claustrophobia. Now, the major, the sort of increase that we've seen throughout the years in the quality of fMRI has, for a large part, been driven by an increase in what we call magnetic field strength. That's the strength of that main magnet that you're lying inside of. So a normal clinical scanner nowadays is 1.5 Tesla. It looks a bit like this. They go to 3 Teslas now. So that's a standard sort of clinical scanner. But nowadays we're increasing the field strength, and the higher the field strength, the better the images are that you can get. But think of it like this is like a, I don't know, like a Toyota sort of car, right? This sort of scanner, they're really in development, right? They're like a hypercar where if you don't know how to drive really fast, you are going to crash. And that's also what happens with these types of scanners that are more like in development. It's very easy to make the scanner crash or just end up with a rubbish experiment, just because there's so many settings that have to be just right. So Nikos is going to tell you all about how we use this giant magnet and the radio transmitters and receivers that are associated with it to produce images. It's really fucking magical. Like, how do you like put someone in a magnet and then create these images, right? In so many different ways. It's really beautiful. So Nikos will tell us all about that. And just one thing, magnets are dangerous. Not the normal fridge magnets, they're not that strong. But these magnets are so strong that they're something like 6 orders of magnitude stronger than the Earth magnetic field. And this also means that whenever you bring something close to the scanner that's made of iron, for example, it will be a projectile that gets drawn into the scanner with like super high speeds. And every year people die because of this. Yeah, it's always good to say that right before the break. Yeah, we'll continue later on, just trying to show you a bit of what is the type of thing that we're looking at when we put someone in the scanner and we get out this data, just to give you an idea of that.
Yes. Yeah.
Yeah.
Oh. Oh, yeah. Yeah, and propose something like Yeah. Yeah. So that's really not.
That's one. The other one. I said, these, you never lose all the. Just know. This one. Voor de per andere vragen hadden bij Brussel, moeten we daar ook echt comments en vragen zetten voor de cijfer? Dan is het gewoon deze.
Hij houdt bij waar je bent in de tekst.
Ja.
Dus daar zijn dingen op gebaseerd. Je krijgt ook een soort van participation score. Dus als je de vraag stelt, antwoord.
Oh, dus dat telt wel mee.
Ja, je kunt een soort van bonuspunten krijgen.
Oké, ja. Alright.
Okay, let's restart. I have so much to tell you. Oh, so just for me to get an idea, like who of you are AI master students? The cognitive science track, is that the cognitive science track? That was an elective. Okay, excellent. And so there's also research master psychology. I hear, yay. Excellent. Anyone else like biomedical sciences? I heard some people are doing bioinformatics. Excellent, excellent. Okay, so this is a very diverse crowd, and I hope to be able to cater to each and every one of you. But the different student populations do have different backgrounds, as you can sort of distill from the different hands, right? So some of you might be really interested in, like, what are the difference between, schizophrenic patients and non-schizophrenic patients in terms of their affective brain responses, affective brain responses. We'll get to that. Yeah, so we'll get to how would you use the MRI scanner to perform an experiment where you can dissociate between the brain responses in these different groups. Yeah. On the other hand, you might be interested, for coming from a AI type perspective, you might be interested in, like, okay, but how is the brain like a metaphor for AI methods, right? So what happens when you take GPT-3, for example, and look at its activation, right, over time, and try to relate that to brain responses that you measure using fMRI, right? It's a totally different type of experiment that you would do. And those types of experiments are also done, and I'm hoping that we will also give you a taste of that type of approach. Yeah. This is really a course where we try to give you the basics and then give you a bit of a taste for all of these different methodologies, so that you can take what you learn here and bring it into an internship, for example. Yeah. Yeah. So, I'm going to sort of show you a bunch of aspects of fMRI data that, again, will give you an idea of the caveats involved, right? And so I'm just going to keep trying to throw bricks at my own field, because I think it's necessary. I think there's a lot of magical thinking involved, and we'll try to deconstruct the thing so that we can reconstruct it with a better understanding. Yeah. But first, when you look at an MRI image, what are you looking at? Yeah. Well, so the really MR physics basics, what are you looking at? We'll learn from Nikos. For now, I want you to know that you're looking at the same type of thing that is a photograph in your iPhone or whatever. It's a rectangular array of values, right? In a picture, those are called pixels for picture elements, right? In a brain, when we do a 3D image, these are not picture elements, but volume elements. And I have no idea where the X came from, but it sounds cool, so we call it voxels. Yeah, so whenever I say voxel, what I mean is this tiny cube of value that's given a certain value, and that is at a specific location in space. Yeah, and it has neighbors on all sides, and together they make up a three-dimensional image. Right? We have three dimensions, X, Y, and Z. Now, This is also something that people use like in hospitals and you know, doctors, they like fancy names, right? And so there are specific names for looking at these 3D images. Like if you're looking at a specific direction, it has a name. And I don't specifically like this sort of rote learning, but it's important for you to know these different directions, right? So what you see here is a sagittal view. And there are a few sort of what's the English word for ezelsbrug? Mnemonic, yeah, like a mnemonic thing. Okay. So how do I remember the word sagittal, what it stands for? Well, you know the zodiac sign Sagittarius? Yes? Same word, right? So what the Sagittarius does is it's the bowman, right? So he does this, right? So what does he do when he stretches the bow? He cuts his own head like this, right? So you're looking in that direction. That's sagittal. Yeah. Okay. So coronal, that's this. It happens when you cut the head like so, right? So in this plane. That's called coronal. Coronal for, well, we just came from a corona pandemic, right? It's a crown, right? And so you would see the crown easily here, right? You would always see the crown. That's sort of my mnemonic, whatever, right? Think of a crown. That's when you see it. That's where you always cut through the crown. Yeah. Now, axial is when you cut like so, right? That's when you slide across the main axis of the human body. Yeah. That's why we call it axial. Yeah. Okay, I won't make a big deal of other things in this way. Okay, so those are the different directions. Remember those because people in general in the literature will refer to specific viewpoints with these words. Yeah. There's also conventions, right? This is called X, this is called Y, this is called Z. They're always the same. There's a thing about neurologists, like doctors, neurologists, being weird relative to the rest of the world. I'm not going to go into that. You can ask me later. It's a story. Normally what you do, and you go into the doctor and you say, I have a headache, they'll make an MRI of your head. And what the usual resolution of the scan like this is 1 millimeter. So what that means is all of these voxels are squares, or cubes, I mean, and they're all 1 millimeter in size. Yeah, that's sort of standard resolution for a whole brain anatomical scan. And we can use the scanner for different, to scan different types of images. And here you see two examples. Here is a T2 weighted scan, and here is a T1 weighted scan. What those numbers mean, T1 and T2, Nikos will tell you. This is just a nice example of how using the scanner in just a slightly different way. Each of these costs 6 minutes to acquire. This is my head again. 6 minutes to acquire each. You don't really hear much of a difference at all when you're in the scanner. It's just that the image that comes out is quite different. Yeah, it's just the way that you use the scanner has changed. Now, if we move to like a 7 Tesla scanner or a 9.4 Tesla scanner, what we have is a stronger magnet that allows us to resolve a higher level of spatial detail. And so in the same period of time, we can acquire a brain with a higher level of detail. And there's all sorts of pitfalls, but what we can do is we can decrease this voxel size, make the measurement more precise. And I'll show you a bunch of examples where we have scanned half of my brain at 0.3 millimeters. So that's 300 microns. That's already at the limit of what you can get with a non-dead person. I'm very trained at lying still, so that you can acquire like 45 minutes of data for one image, right? But you know, the things become problematic. So one thing to realize though is that it doesn't sound like much. This is like, oh, it's factor 3, right? It's 3 times more precise, you would think. Yeah, but the signal that we capture comes from the protons, right? The protons, the amount of protons that are in a specific location, it depends on the size of the cube that you're measuring from, right? So it doesn't mean that you're 3 times smaller voxels if you go to 0.3 millimeters. It means 3 times 3 times 3 smaller, right? So 27 of these 0.3 millimeter cubes would fit into a 1 millimeter cube. Yeah, that also means that it becomes 27 times harder to scan. Yeah? That's really what we're really bumping into as we're trying to increase the resolution, right? So decrease this voxel size. We are sampling smaller and smaller types of patches of tissue.
So is that also important?
Yes. Yeah, it's yeah, so this is something that I'll show you in a bit. It's even where so you're measuring veins, right? And you're very sensitive to movement. And everything that you want, as in more detail and faster and all that, there's more problems whenever you try to do something nicer. So you can have nice things, but it's always a lot of work. There's no free lunch there. Yeah. Yeah, so just as an example, I just showed you this T1 and T2. Nikos will tell you about what the difference of those two is. But the scanner is really, really versatile. It's pretty cool that you can just put someone in the scanner, measure their chemical, so the chemical composition of certain patches in their brain. You can do this anatomical scan. You can also, and I'm showing you an example here, right? So you can add dimensions. So for every voxel, you can measure not just over time where you're measuring fMRI, and I'll show you more about that in a bit, but you can also measure the orientation of fiber bundles inside each of those voxels, right? So what I'm showing you here is a reconstruction of fiber bundle directions measured at each of these voxels. And you can then trace the tracks running through the white matter of the brain. So the white matter contains all of these sort of highways of the brain. I'll talk more about that in the neuroscience lecture later on. Yeah.
Is the voxel, does the MRI take a voxel every interval, or is it like all in one go?
Yeah, so that depends on how you're using the scanner. So certain types of data that you acquire, they will be actually per slice. And so you acquire like an image per slice. You do run through what you might call pixels, but they are not pixels. You get into sort of the mathsy, physicsy side of things. It's about how you manage to create, how you can encode space using just frequencies of radio waves. That's really the trick that happens here. So you can also, you can already feel how physicsy that must be. And so you're not sampling voxels specifically at specific moments in time. And it really depends on how you use the scanner, what the specifics of that type of sampling is. So we'll get into that more. Excellent question. And it's like Facebook statuses, it's complicated, right? It's really complicated, but like need a physics PhD complicated sort of thing. It's yeah, most people are better at relationships, I think. And so you can also, so here this measures the direction of diffusivity of water in a specific location, and that allows you to track trace these fiber bundles. This here shows you, this is the heart. You can actually image the velocity of blood flow through the aorta, for example. So the velocity and the direction of blood flow. And so you can use this scanner for all manner of things. And that's really cool because, you know, this means that doctors can use it for all sorts of things, but it also means that we as scientists can use it for all manner of things. Yeah. Yeah.
What's a four-dimensional?
Yeah, that's a good point. I'll get into that in a bit for fMRI. So you have X, Y, and Z for space, right? So that gives you a three-dimensional box. And if that's just an image, then you just have a value at each of those locations, right? And like I said before, if you have functional MRI, you just have multiples of these boxes, right? So then the fourth dimension is time points. Yeah, so if X, Y, Z, T. But for this type of thing, you actually have X, Y, Z, and then a vector, so it becomes a tensor, like you may know from AI, right? Like an n-dimensional tensor that has like the strength of the signal in different directions. So it's encoded as similar as time points, but they're the strength of the diffusivity in different directions that you then can use to create something like this. Here, it would be like the value would be like the strength of the movement of the blood in each of let's say 100 different directions. So you get a fourth dimension in this case. Yeah. And you can imagine, it gets really complicated on the physics side of things. So you can have 5-dimensional and 6-dimensional, and yeah, it can get a bit hairy. But yeah, 3-dimensional images, and then you can add a 4th dimension that can be any manner of things. Yeah. Yeah. Not necessary, no. It's easy to add the time dimension because you just ask someone to be in the scanner for a bit longer. Do the same thing twice, and then you'll have time. So it's not necessary, and many of the anatomy images or these types of images, you just have the single image, and you don't care mainly about the functional aspects, right? So anatomy, we don't think will change on the order of seconds, right? Although there are interesting findings that sort of show this but it's beyond the course, yeah. Does that answer your question? Because I was going off on a tangent, but yeah. Okay, good. Any other questions about this?
Yeah, with the time dimension, you said that one scan takes about 6 minutes. So can you just keep them there and it will scan precisely the entire brain again with another time dimension? Or do you need another 6-minute scan and then you have time dimension of 2?
Yeah, that would be the case. But these, and I'll get into that in the next slide. This is the hallmark signature of an excellent question is if the answer is on the next slide, right? In terms of fMRI, what you do is you acquire this 3D box, but you acquire it not every 6 minutes, but you actually accelerate what you're scanning. And so you try to do this every 2 to 1 to 1.5 seconds. So what you're trying to do is get these snapshots every second, more or less, ballpark. It kind of depends how fine the detail is that you're interested in, how strong the scanner's field is, that sort of thing. So it's not 6 minutes, it's like 300 times faster than that. But of course, you can imagine there's no free lunch, right? The time has to come from somewhere. And where it comes from is space. Yeah. So you sacrifice one, you gain the other. Still the same scanner, but the sequence, you change it to acquire data differently. And so, in terms of fMRI, I'll just take a step back and then you'll see what this means. In terms of fMRI, what you measure is this 4D matrix, right? Similar to that diffusion thing, we have this 4th dimension, and all we have is blood-sensitive anatomical images, just a whole series of them. Yeah, so it's very simple, really. So, like I said, you want to scan this fast, so you lower the resolution, you increase the size of these voxels that you are working with. And so instead of these anatomies that are usually at that scale, about 1 millimeter or smaller, where it takes 6 minutes, you can go to like 1.5, up to 4 millimeters in size. And again, the larger the voxels, the faster you can scan, right? So if I go to the 70 and I say, I want a 1.5 millimeter whole brain acquisition, we say, then you can get like a frame time, a repeat every 1.5 to 2 seconds. If you go to 4 millimeters, right, that's again the cubic relation, right? You can go to 200 milliseconds or so, right? So you can go really, really fast if you're interested in that sort of thing. And then this is that fundamental trade-off. The higher the resolution, the smaller the voxels that you're interested in, the finer the level of detail that you want to have in your functional measurements, the longer it will take to acquire those data. And so what Nikos will tell you all about is how that is determined by the physics, but this is the rule of thumb. Now, relative to these anatomical images, we're scanning really fast. Like I just said, like 300 times faster, right? And this has to come from somewhere. It means that not only do we get lower resolution images, we get sloppy images. And the next 10 minutes is going to be me showing you how fucking sloppy fMRI data is, right? And I'm going to show you a beautiful data set even, but it's sloppy. So what that means is we get a lot of noise, we get a lot of artifacts, and this is really caveat number 3. When you see these beautiful blobs in the brain, and they say this is the love region in the brain, where it comes from is a bucket of noise that they have massaged into producing a nice color figure. Yeah. And so what this course will do is show you how you could create those figures, but you have to be aware that this is always coming from a bucket of noise. Yeah. It's a nice and square bucket, but it's a bucket of noise. Okay, so what does that look like? It looks like this. This is a movie, again, of my brain, and this is a beautiful data set. And it's just a movie. I'm sitting in the scanner. I'm looking at, I don't know what I was looking at. The signals that you get from, for example, viewing a movie, they're so small that you will not see them on a scan like this. Yeah? What you do see on a scan like this is me just traversing the scan, right? This is not an artifact or whatever. This is me just going through. You can see the mouse pointer here somewhere. And I'm going back. What you see is that there's a lot of shit happening in the middle of the brain. It's the heartbeat. You see the heart pumping, like pumping the blood into the brain. And that's the signals that you see. You also see that, especially at these corners of the brain where there's like a lot of interaction between air and the tissue, you get all sorts of weird artifacts. It gets warped. There's like a hole here. I don't have that hole in my frontal lobe. It's an artifact of the image. You see these sort of ripples in the image. It's because we are pushing the scanner to acquire data really fast. That then gives us these ripples, this extra noise, this dropout, all of that, right? Yeah.
So what kind of resolution are these photos distributed?
Yeah. So I'll get into that in a bit. So again, yeah. So a lot of noise. You see how noisy this is, right? It's a nice image. This is actually a beautiful image. Like I said, this is a beautiful example data set. But yeah, it's still very, very noisy. Now you can make it look nicer. So what I've just done is I've just changed the way that the viewing program shows me the data, and it just interpolates things. It smooths things out. It looks much nicer, doesn't it? Still, you see that when we go through it, you start to see these ripples, especially if we go down further, which I think I'm doing. You start to see this hole here. See these ripples here? They don't exist. It's all the image artifact, right? So you have to be aware that even though the previous one was very blocky and this one looks really smooth, exactly the same data, I'm just changing the way that I'm showing it. Yeah, it's just a beautification thing. Yeah.
So how long does it take to find it?
Yeah. So here, every image is taken 1.6 seconds. Yeah. And I think I can go to the next. Yeah. So here, the original data, which is the high resolution, the fine detail data, is a very exquisite 1.7 millimeters for every voxel. So it's small for a functional imaging thing. And this is around this for us, the sweet spot for a whole brain acquisition, we can do like around 1.5 seconds, a little more than 1.5 millimeters. So you get a lot of spatial detail and you get a reasonable temporal resolution to perform functional MRI analyses on. Yeah. What you see here is that I'm changing the resolution from 1.7 to 3.4. And 3.4 used to be the sort of standard resolution 10, 15 years ago. Yeah. And I don't know if did you see it go back and forth? I think this movie is a bit shorter or something. Maybe I don't think. Oh man, there we go. I think. Yeah, so this is me changing the resolution to 3.4. So this is what we're working with now. This is what we used to be working with, right? You see that we used to be working with even worse shit, right? Great, that's clear, right? We have beautiful images. They still don't have really the resolution and the fine detail that we would have in an anatomical scan, but at least we have it every 1.6 seconds or so. And this is acquired at a 7 Tesla scanner. You cannot get this level of detail at a 3 Tesla scanner. Yeah. Sorry, Rebecca. Yeah.
Is the upscaling methods with AI used?
Yes, that is an active area of research at the Spinoza Center. We are actually doing things like compressed sensing. I don't know if you're sort of familiar with that sort of technique, but you can, in principle, make very sparse measurements. So you don't have to record every voxel. I'm speaking in metaphor here, but you don't have to record everything. You can record very sparsely, and then what we call reconstruction. That gives you a very nice image, even though you haven't actually acquired all the data. So you use a neural network to impute a lot of the information. Yeah, that's what people are actively developing. Yeah, so scanner, sort of the companies that make the scanners like Philips or Siemens, they're actively developing this sort of thing because it means that you can scan people 3 times as fast, right? More patients, more money. that sort of thing. Yeah, so it's a very active area of development. Okay. Now, I want to show you a bit of, I'm going to go a bit technical maybe for a first lecture, but I thought it'd be good to just focus a bit on how bad data are. Because I don't know, it's sort of a masochism thing. Like I'm supposed to like sell this field, right? But I'm not really. In any case, I just want you to be aware of how crappy the data is that we work with, because then you can appreciate how beautiful it is, the types of things that we find using fMRI later on in the course. Yeah? So what we care about is signals, right? We want to get signals from the brain, something, even though it's blood, is going to relate to the neural activation so that we can actually look at the machinery that produces our minds inside a living human brain. Right? That's what we want. Those are signals that we're interested in, even though it's blood. But like I just showed you, there's a lot of noise, right? There is heartbeat stuff happening. Right? I don't want that. It has to go. Right? And so our eternal battle in analyzing fMRI data is to separate the signal from the noise. Yeah? To be able to say, this is noise, I don't want it. This is signal, what does it look like? Yeah? But of course, no one knows where the boundary between signal and noise is. But we have standard ways of quantifying the quality of data, right? So we want to have high signal and low noise. And so our standard measure for scan quality that you will see appearing also when we look at pre-processing and that sort of thing is called temporal signal to noise ratio. And so when people say TSNR, that's what they mean. It's a very simple measure because it's just if you have 100 images like the one that I showed you just now, you just take the average. You average over the time dimension and you end up with not a 4-dimensional, but 3-dimensional data set. And in the first practical, we perform that type of operation. So you're asked to produce a TSNR image. So that's the mean image. And of course, you can also, over that 100 images, you can take for every voxel the standard deviation. Yeah, that's the amount of variability that you have along that axis in the image. And so that gives you the standard deviation over time, and you just divide one by the other. It'll become clear why we have to do this sort of thing, but you can imagine that a very good image has very high signal to noise, right? The mean represents the signal here, and the standard deviation represents the noise. So this ratio expresses signal quality. And so just to give you a ballpark idea, right? If you have a normal fMRI scan, if you are looking at the air, it should be close to 0 because the mean would be close to 0. If you have noisy gray matter from like a 3T experiment, it will be around 40. But if you go to 70 and have large voxels still, you get beautiful signal, and then you get something like 80. And white matter usually doesn't have any variation because it's just there. It doesn't have the cell bodies, the blood vessels to respond. And so that has usually pretty high SNR. So what I'm showing you here is a standard deviation image, and I'm just going to zoom through it a bit. I think if I click, pretty sure that if I do, yeah. So this is a standard deviation image. This is where the variability in an fMRI time series is, right? So all I've done is I've taken this 4-dimensional data set and I've calculated the standard deviation over the time dimension. It gives me a 3-dimensional image, right? One number standard deviation. And I can just look at it. And what you see is that the variation is exactly where we saw the variation be in that movie already, right? You see that in the center of the brain, you get very strong standard deviation, a very high standard deviation. That's where the blood was pulsating. So this gives you a very nice indication of where signals are good and where signals are bad. You see that there's a lot of variation in the gray matter and even more on the outside, because whenever you move, you're taking a bit of air, for example, and you're moving a bit of brain in there. There's a huge fluctuation in the signal in that voxel. So that's going to give you a lot of standard deviation around. So translating this into what we call this TSNR image, that gives you something like this. Of course, there's no signal in the background, so we can really zoom into the brain. So what you see here is that we could, for example, just take all of the voxels in the frontal cortex and calculate the TSNR in the frontal cortex. And thereby we could compare across different scanners, how good is the data, right? That's really why we use this measure of TSNR. Is that clear? It's a very simple measure. And we don't even care about activations or whatever, because they're tiny compared to whatever you're seeing here. Cool. Now, caveat number 4, right? when we see these fMRI images as four-dimensional data, we can visualize them. In that first practical, the end of the first practical, you'll be visualizing some of these types of images, right? So you can just write a couple of lines of Python code, enter, you see this type of image. Now you can sort of zoom through it. But it's important to realize that when you do this, you have a four-dimensional matrix in some memory in the computer. This is values, right? And other than other scientific measurements, values that come out of an MRI scanner in general, and specifically in terms of fMRI, mean nothing. They do not exist as SI units. So normally when you do a scientific measurement, you would say, the velocity of this car is X meters per second, right? The meter and the second are standard scientific units that mean something, right? And if I measure that speed in different locations in the world, and I'm not a physicist interested in relativity, then I can compare these things, right? Meters per second is meters per second, yeah? Same goes with temperature. All of these units that we use in science, they are fixed, and then that means that we can actually compare them across experiments. No such thing in most MRI measurements. We can be very careful about our MRI measurements and then actually produce things that are calibrated in this way, but the standard fMRI images that we get are unitless. They are just numbers. And you can imagine that you don't have any firm footing anymore when you have only numbers, right? How are you going to relate from scanner to scanner? Well, you can't. And this is one of that caveat number 4. I should have made that explicit. We cannot directly compare these things. And so we have to use shorthands, right? And you see that this whole field is just, oh, we have this thing, we can do stuff. Well, let's see. Oh, this doesn't work. Oh, let's do this sort of thing. You really see how new a field it is. Now we have to find a way to compare across scanners. You'll see that we can do that using statistics later on in the course. But in general, what we do is if we want to focus on functional MRI signal strength, we use a shorthand, right? Knowing that we are looking at something that is unitless, what we can do is express things in percentage signal change. It's related to the TSNR, that's why I put it here. Because you're using more or less the same thing. You're using the mean image and you're just dividing by it, which gives you the fraction of the response relative to the mean image. And then you multiply it by 100 to get a percentage. That's it. So you can get huge signal fluctuations in these brainstem regions, right? Where the blood is pulsating really strongly, you get huge fluctuations, right? But they are much larger than so it's very easy to get 40% signal change by headbanging in the scanner. That's not really what you're interested in. That would fall under the NOMER noise, right? What we are interested in is functional signals that tie to neural activations through this blood response. Yeah. And so if we're interested in this in terms of percent signal change, what are we looking at? Well, normal values for this sort of thing are, if you're conducting a normal experiment at 3T, you are looking at percentages up to 1%. Right? So relative to the mean image, to the anatomical image that you're getting, you're looking at 1% fluctuations over time, right? It's fucking tiny, right? And even like if you're looking at frontal lobe and things where you don't really know what the signal would look like, this really goes down to 0.2. So in the literature, you will see 0.2% signal change values regularly. And it's not a problem if all of your subjects show it in exactly the same location and that sort of thing, right? Statistics. But these are tiny values relative to the actual images that you're looking at, right? So important to realize that that's the case. Now, if you have a very well-defined region that you know how to make a very, very strong response there, and this is the case in primary regions in the brain, so primary motor cortex, primary visual cortex, you can really pound those regions and make sure that all of the neurons there are firing. So for example, tapping your finger really, really fast, where I can't even do it, that would give you like a 3% signal change, right? So that's the maximal response that you more or less can get. There are theoretical limits also, but this is in practice the limit that you can get. So if you go to 70, you might go up to 7% signal change. That's enormous, right? Relative to what people usually get. And then you can have these problems where you might think, I have the most beautiful voxel in the world. It's 20, 30% signal change. Now let's look at that equation. How can you get 20-30% signal change? Well, you can get that level if your mean image is small, right? You're dividing by a smaller number that blows up the resulting value. So this happens when you're looking at a vein. And veins turn out to be really dark in the functional data that we, so you have a very dark voxel, very low mean value, you divide, and you see this huge increase. We're not interested in these veins, right? This is just purely blood. It's no longer related to neuronal activation, we're just looking at a blood response there. So we don't want these large veins that we're sampling from directly. So just to show you again, me zooming through my own brain here, veins, for example, this part, huge vein, very dark. You see that? So you can imagine that if that value was already low and you divide by that mean image, it's very easy to get huge fluctuations. Yeah? So just to highlight a few veins, here's a vein, those are veins, and I'm going to zoom through, see veins here, that's a vein, all veins, right? Thing is, you don't actually see these veins if you're scanning at a lower resolution. If your voxels are larger, you're going to have some gray matter, some vein, right? So so everything blurs together a bit. So these are such good data. Oh yeah, so here's breathing response near the, so you see these fluctuations? That's me breathing. Right, so you see not only the heartbeat, but also the breathing gives you these signals that you're not interested in. Now I was looking at a very specific response, or looking for a very specific response here. Just clicking around the brain, I only see noise, right? It's breathing, it might be movement, it might be whatever. There's one thing that I was interested in, and that was this flickering bar that was on the screen 8 times during this experiment. I think it was, maybe 9, I forget. Yeah. And this is just here to give you an idea of the percent signal change that we might experience, right? You've seen all of this noise, right? The time courses that you see here at the bottom, I hope that was clear, I forgot to say. But it was clear that what we're looking at is for every voxel that I select here, we're looking at a time course, yeah? Yeah. Okay, good. And so this is like the most beautiful response voxel in the brain that I could find, just making this video. And you see that you get these very nice responses, right? This looks really like there's a signal there, right? We're doing something to the brain and it's responding according to the experiment that we're conducting. Yeah? And so this is really what we're trying to find. We have an idea of what the signal should look like, and so we can try to find it in this bucket of noise that you see at the top. And that's really what all the experimental analyses, or all the analyses that we'll be teaching you are all about. So what you see here is this is 13% signal change. I was looking at a vein. I was so happy when I found this voxel because I thought it was beautiful. It's just a big vein, right? And it's likely this vein, and we'll go into the neuro part of that a bit more, like the anatomy part. But if you're sampling a vein, it's likely that it will just be pooling blood from like a huge region into that vein. And so you're not looking at any specific measurement anymore. So you get a huge response, but it's not really the type of thing that's directly linked to neurons anymore. You see how that link between neuronal activation and blood is indirect. And if we don't understand what we're looking at specifically, then we can misconclude. So this is, again, a caveat. Yeah. This is actually a repeat of caveat 2, I think, but yeah. Okay, so you get an idea, right? A bucket of noise. I hope I can still sort of regenerate some enthusiasm later on. Because just to be clear, fMRI is fucking awesome too. But we'll get to that. So this you might call signal. This is at least the type of signature that you'd be looking at, right? These responses, they look completely different from all this movement and cardiac related noise that was going all over the place. It's definitely also not the movement or breathing related stuff that we might not be interested in. But this really looks like signal already, right? You would agree, right? This is the type of thing that might be something that we're looking at, or we're looking for. Okay, so yeah. How do you go about this, right? Now you have an idea, like we're looking at a bucket of noise, and our goal from most of this course is really to show you how do you take this bucket of noise and produce something that you can say, I'm pretty confident this and this was happening in terms of neural activation. Through all of these different steps of uncertainty that are associated with it. So how do we do that? Well, we need to make an experiment, yeah? And we have to keep in mind that we're looking at this whole sort of pipeline of analyses where all of the different choices that you might make are interacting with one another. But here's a schematic that I think is interesting, or at least shows you a bit of the different things that we need to be thinking of when we do our experiments. So one thing is the most important. Whenever you do an experiment, be clear about what your goal is. If you're just measuring stuff, you're fucked, right? Just measuring stuff isn't really science, it's hobby. I do it a lot, so I'm guilty, but you always need to be very aware of what your goal is, because your goal will inform all of the choices that you make. And if you let go of your goal, there are more ways of going wrong than if you have a clear idea of what your goal is. Yeah? So you want you want to be very careful about how you acquire data, right? Put people in the scanner in the right way, choose the sequences that you use, the different scans that you make in exactly the right way. They have to be targeted in exactly the right way. Yeah. If you are interested in something that's going really fast, you shouldn't use really small voxels because they take a long time to scan, for example, right? You shouldn't accidentally only scan frontal lobe if you're interested in the visual system, which is at the back of the brain, and you're not scanning that. Yeah. So there's all these choices that you make. That was a bit silly, but you get the point. Now, we'll have a complete module about pre-processing, because when we get this bucket of noise from the scanner, we have to do all sorts of things. People move slightly inside the scanner. We have to sort of realign their brain so that whenever we say this is voxel X, we're actually certain that it's at a specific location in their brain and not just something that's drifting through space because people are moving, right? So motion, we call this motion correction, is very important. Now, these data can also have very strong drifts just because people are like, falling asleep usually slowly. And so we want to get rid of those very slow drifts because we're interested. You saw that signal, it's going up like so. Right? That's the type of thing that we're interested in, not these very slow, not that slow stuff. We also want to acquire an anatomy so that we know for every functional voxel, where is it in relation to the anatomy? That means that we have to perform a co-registration step between the functional data and the anatomical data, right? So we have more detail in terms of where things are. And like I showed you, there was a hole in my frontal cortex. These fast fMRI images, they are warped. When we say that something is somewhere in the brain, according to one of these images, it has image-related warping artifacts that we need to correct for. So we'll also go into that, how that works. Yeah? So a lot of pre-processing that we have to stack on top of one another in order to be able to perform the analysis that we want, right? To get the data where we can actually start saying, this is signal, this is noise. Because without doing all of these steps, we're lost anyway. Yeah. Now, although that sounds really negative, I'm very sorry about that. It's going to be awesome too. In any case, so there's different types of experiments that you might run. You might want to localize something, like I said, like the love region of the brain, right? So that's when you're just interested in localizing, like this bunch of voxels activates when people see a picture of a loved one instead of, I don't know, a robot or something, right? So that's the type of thing that you might be interested in. That's a very classic type of experiment to conduct that people are less interested in nowadays. You can look at how different brain regions are connected in terms of their bold responses, in terms of fMRI. You can try to map how different voxels represent information. That is a much more modern way of going about things. And you can try to do things like predict from someone's ongoing brain activations the type of choices that we make. So really trying to understand what the pattern of brain activation in a region, for example, means, and then trying to predict behavior. Now that sort of thing, these lower regions of this thing here, they are far more modern ways of going about fMRI. And this localization thing is a bit of old school sort of thing. Yeah. Yeah. So yeah, it's kind of a vague term, right? But for example, what you could do is you could see how the response in a given voxel correlates with or like aligns with the responses of some neural network, right? And so then you'd have all of these nodes in your neural network, and you map how this voxel sort of aligns with all of these different nodes. And there might be a single node, I'm caricaturing here, but there might be a single node in your neural network that corresponds best to this voxel, right? And so you have this sort of information mapping, because now you have some model of what this voxel is doing, right? This voxel responds as if it's this part of a neural network. And so that type of information mapping that I'll show you a lot of examples about later on in the lectures, But that's the type of thing that we're interested in. So we can create, for example, semantic maps of information processing throughout the brain, that sort of thing. Yeah. I think I have still 3 minutes, so I'm going to try to speed up a bit. If you have any questions after the lectures, you can come to me, and I can also, there's specific channels in Discord for the different lectures. So you can ask me specific questions there. Also, yeah, because I'm now going to have to like speed things up a bit. Cool. So I showed you all of those different pre-processing steps that we need to do. It gets more complicated than that, but just to get our data into an analyzable format, we need to take all these steps. And what this means is that people used to be able to like tinker a lot. Like, oh, I'm not getting the result that I want. Well, let me just change my motion correction a bit. Let me just change my warping a bit. Right, so people would be able to tinker their analyses, and this of course just wreaks havoc on reproducibility, right? It means that you can have all of these non-reproducible practices in your research, and this is also why fMRI has this reproducibility crisis that we'll get into. Now, our goal is to teach you novel methods, like modern methods, that really approach science as a sort of open thing, that your entire processing pipeline should be open, that your data formats should be open so that you can easily share information, and that you cannot fool people either through tinkering or by just being sloppy, because we're all sloppy. Yeah. So what we're trying to teach you during this course is also like how to adhere to those data processing formats, data saving formats, and so forth and so forth. Yeah. This means you can very easily open source your data, share your data with other researchers, and also just take other people's data. So there are huge repositories online where you can just get people's data. What I'm showing you here is examples of a standardized pre-processing pipeline, for example, that we'll be diving into in the pre-processing lecture. Now, I already showed you that fMRI is getting better in terms of temporal resolution, in terms of spatial resolution, right? We're improving more detail, more speed. One thing that people are trying to do more, especially at these 7 Tesla scanners, is to look at the different lamina of the brain. So we know that the cerebral cortex, we'll get into this more in neuroscience and further lectures, but we know that the cortex consists of these laminae, like different levels of processing is what they stand for, and we can actually start measuring those. Another really modern thing is that we have these very large open science projects, and you can just download the data. So examples of that are things like Human Connectome Project. I've used that to great effect. Like 1,200 subjects. Your laptop won't be able to contain that much data, but if you have a good server or something, you can start using it. This UK Biobank is insane. Like, I think I'm missing a 0 here. Now it's at 100,000 subjects. And they have like genetics and all of that in there. So huge open data sets if you're interested in that sort of thing. It's really gorgeous. And another example is this natural scenes data set, where they didn't do many subjects, but they did do a shit ton of scanning per subject. So each of these images, each of these subjects was shown 40,000 images in the scanner. So you can do like a ImageNet type of thing, but then on brain activations with this amount of data. That was the goal for this data set. And so one of the reviews that you'll be reading later on is really like how to think about this trade-off between number of subjects and scanning time per subject, and depending on, again, what your goal is in terms of research. Yeah. And we'll focus a lot on things like open source software. We'll be using open source software also in the course. Oh, is it? Yeah. And all of the software that we'll use is, you can also get it to be containerized, reproducibility. It's a really big thing in the field. Yeah. I think I'm almost there. So we want to start measuring like even more detail. So what I'm showing you here is a functional acquisition at 0.7 millimeters. So you can really try to push the scanner and get really high resolution data. And you're starting to enter this, you're sort of shifting that box or expanding that box into the territory where it starts to overlap with things like non-human electrophysiology, right? So you can start to compare findings across different species of animals, us being one of those species, for example. Yeah. Yeah, so one thing that I think many of you will be interested in if you're coming from the cognition track of the AI masters, for example, is how do neural network architectures, for example, align with brain activations? Yeah. And so we're going to get into that. So this is that information mapping that I was talking about. I definitely recommend going to this URL here just to see a demo of the type of thing that people can produce using this type of approach. Really looking at how the brain represents, in this case, semantic information. It's a gorgeous study, and we'll get into how they did this research in later on lectures, but it's really cool to look at. And this idea of being able to look at how different brain regions communicate with one another through what we call functional connectivity is also really one of the major things that are developing right now. So we can look at things like connectivity measures between different regions there. Okay, so that's it. And start watching the YouTube videos. They give you a bit of background. The YouTube videos are from 2015, so they're old. And so they are very much focused on this localization thing, and they're doing a lot of statistics that are like traditional. My focus will not be on things like traditional statistics, because I want to be able to show you like novel things also. But watch the YouTube videos, they will make things clearer too. Yeah. I'm looking forward to this. We're gonna have fun.